Okay, everybody, well, I think we'll start with the seminar now. Welcome to all of those who are on VideoCon as well. Thanks for joining us. So our speaker today in the University of Washington Astrobiology seminar series is Professor Janet Siefert. She is an evolutionary molecular biologist, to get that right. She got her PhD at the University of Houston, where she worked with George Fox as her advisor. And George, as some of you know, was the discoverer of the archaea. Janet is now currently working as a faculty fellow at Rice University in Houston. And her research interests include theoretical biology, horizontal gene transfer, and microbial ecology. And today she's going to be giving us a talk entitled Stromatolites, What Sulfur Got to Do with It.
Well, I'm glad to be here. You're going to do the live? I'm going to try to, yeah. Okay. Okay, I'm in the dark. Can't see me. That's good. You'll have to forgive my southern accent. I'm from Arkansas, so I try to hide it a lot, but it'll come out every once in a while. So if you don't understand something I'm talking about, well, just raise your hand, and I certainly don't mind being stopped. How many of you are astronomers in here? Biologists? Cool. Oh, see, you told me a lie. You told me there were going to be nothing but astronomers in here. Okay, so I'm going to tell you about, actually I'm going to tell you a little bit about one of my favorite places on Earth, and I'm going to tell you about some research that's being done there. And the way I'm going to introduce it is I'm going to talk about the stromatolites that are there. And the first slide is just my slide of recognition of everyone that's working down there. This is quite a group. Being a part of the astrobiology, you understand that it takes a huge number of people to actually tackle some of the problems that we try to tackle today. And that's reflected in the number of collaborators that I have here. I won't go through and name each of them, but they're across the border, across the Rio Grande, all the way through the southern part of the United States, and of course, very appreciative to Vicki. All right, so let's get started. This is a picture of early Earth. I know you've all seen it. It's pretty good that we got this photograph from somebody. There's a very clear feature in this picture. Of course, this is an artist's rendition of the early Archean on the Earth. But as you can see, there's some features right here. And these look disturbingly like current features that you find off the coast in Australia in Shark's Bay. Those are stromatolites. So a stromatolite is actually a specific... That's okay. You can't see.
There we go. That's okay. Sorry.
I thought he was going to have me do disco in a minute. All right. So these are stromatolites. And that's a very specific name for a kind of feature that microbes make. And the general term for that is a microbialite. So basically, I'm going to be using the word microbialite, but most geologists and most people that are familiar with these think about stromatolites. And the difference between the microbialite is the general term. The stromatolite is a specific description of a kind of microbialite that has layers to it. Okay? So turns out I don't know why, but it's sexier to say stromatolite than microbialite. So everybody calls them stromatolites, but the general term is microbialite, and that's the one I'm going to be using for the most of this talk. So here, that was what they looked like 3.5 billion years ago. Here's what they look like today. These are in the Bahamas. Clearly, these are at least 5.5, 6 foot tall. These are at Shark's Bay, Australia. And almost always, if you find microbialites or stromatolites in today's ecosystems, it's because there's something extreme about that environment that's allowed them to actually grow. All right. So what we're going to talk about is how you actually form microbialites. So the recipe for them is that it's really, in essence, just a simple chemical precipitation. The chemicals in the water, the carbonates in the water, are actually precipitating around the microbial community. That's what's happening. It's the same process that happens in coral. It's just a different organism that's actually doing it. And it's always about the community. There's something about the microbial community that's making that precipitation occur. We're going to talk a little bit about why sometimes you have microbial mats, but sometimes you have actual microbialites. For some reason, these things do not cause the precipitation, but when you get a microbialite, the precipitation has actually occurred. And today, in today's world, just what I said earlier, you must have some sort of extreme environment to allow that to happen. Now, let me explain that in a little bit more detail. Back in the early Earth, when all you had was simple life, microbial life, you didn't have the natural set of predators that might eat the microbes that would allow them to be not robust and not allow the reactions to happen to cause the carbonates to fall out on top of them. Today, we have predators that eat the microbes, snails and fish and different things. So unless you have some way of keeping those predators away from the microbial community that can make the lithifying mat, you're not going to find them. So anywhere you find microbialites or stromatolites in today's ecosystems, it's because you kept the predators away somehow. In the Shark's Bay, Australia, it's too salty for the fish to come in and actually eat. So that's why you have them there. Okay.
Excuse me, stronger question.
Sure.
I've heard this before, that, there's all grazing today, but I mean, there's a lot of grass in the world today also. We get grazing on it, so that's not sufficient in itself to explain the lack.
Okay, that's a good question.
An equilibrium is established. You're saying the equilibrium for some reason is if you don't have the extreme conditions.
Right. Okay, so as usual, that is an excellent question, and I've never actually thought about it from that context. I mean, you make a good point. But what I'm fixing to show you about a place that actually has these stromatolites, there is another reason why the environment is so extreme that you don't have the predators actually working on it like they would be. Now, to compare it with grass, I don't think is a fair comparison, because the actual bacteria that are producing these, the predators can actually just keep them cropped down, grazed down enough so that they can't actually produce the chemistry that needs to cause the carbonates to fall out. Whereas, if you're grazing on grass, I mean, you can leave a field fallow or whatever you want to and let the grass grow up or whatever. It's a little bit different situation. But the bottom line is the natural predators, we had a microbially dominated community prior to the rise of oxygen, or prior to 2.5 billion years. That's in the rock record. We have those. And now today we don't have them except in extreme environments. Now I will admit to one thing. I think we're going to find them in more places than we realize. I don't think they're going to be quite as rare as we think they are. That sort of gets at your question a little bit. But it still has to say something about the ecosystem and the community. There is still some reason why they're not just all over the place like they were back in what we think they were in early Earth. Now, I really like it when you ask me questions. That way I know you're not asleep. All right. So what I've tried to tell you is that given this early time in Earth and we don't have all these predators and you've got microbes dominating, why is it that sometimes you have a mat that doesn't cause any kind of precipitation in the chemistry of the water to precipitate around it, and sometimes you do? Well, there was some great work done by Dupraz and Fisher. Dupraz has actually put some excellent papers out on this. And what they did, you don't have to understand too much about this graph. I realize it's pretty busy, this table that came out of their paper. Basically, what they're showing you is that sometimes mats lithify. These are two examples that do. And sometimes they don't. Lithification doesn't occur. If lithification occurs, you're going to be able to find that in the rock record at some time. If it doesn't happen, you won't, there'll be no evidence. It'll just, the soft bodies of the organisms will not be captured. So what causes this to happen? Well, they discovered that there's sort of two processes that are at work. Sometimes you have a metabolism-controlled environment that's causing the carbonates in the water to fall out on it. And basically what's happening is the metabolism around the colony is just changing the pH enough so that the carbonates fall out around it. Or you may have the situation where the community is in such an environment that You've probably been around bacteria in ponds or something, you've seen it, and it's kind of slimy and gooey. Well, there's an extrapolymeric substance that some bacteria put out. It's kind of like a net that keeps everything sort of together and in a mat system. That's EPS, so extrapolymeric substance. If the community is creating that kind of substance around it, and it's in a place where there's actually kind of a washing in of detritus, then that detritus can be trapped in the extrapolymeric substance, and that can create a stromatolite or a microbialite. So this paper is just basically showing you that if you have either one of these situations or some combination of these situations occurring, you're likely to get a lithification process. Whereas if you don't have these two situations happening, you won't have a lithification. Okay. Any questions on that? All right. So now to some real-life microbialites in a really interesting and cool place. This is Texas, which is where I'm from. This is Mexico, and this is the Chihuahuan Desert, one of the great southwestern deserts. It's right in the middle of this, right in the middle of the desert, is an oasis. Now let me show you some pictures here. Vicky's been down here. This is a valley that's up pretty high in the mountains, completely surrounded by mountains, and there's pools that are from subterranean vault system that actually are coming to the surface. There's over about 200 of them. And then you see in this picture, the next picture, they actually look like little pieces of the Caribbean just dropped down into the middle of the desert. There's over 200 of them. You can actually see the fault line running down this one. This one is just about like nice warm bath water. It's that warm. This is an aerial view of some of the ones in the valley. They're called posas. And this, right around the rim here, is a microbialite, that white rim. So these mats, for probably thousands of years, have actually been lithifying in this particular place. Now, a little bit about this place. The area is 1,200 square kilometers. The rainfall is less than 150 millimeters per year, pretty dry, and it's 740 meters above sea level. The other thing that's interesting is there are dunes there that are part of one of the river systems that runs along the surface of the desert floor. And I don't know if you could even hazard a guess what this is. Does this look just like sand dunes to you? This is almost 97% gypsum. They actually mined this for drywall for a long time. So this is carbonate, sulfur carbonates. So there's some special things that are going on in this valley that I'm trying to get you to understand. One thing is it's pretty high up, so the water sources are not coming from anywhere up other than just the subterranean fault system that you have. It's ringed completely by mountains, so it's very isolated. There's a sulfur system, there's a sulfur cycle of some kind because you have all of this gypsum obvious in the topography. And there's almost no rainfall. Okay, so the Cuatro Ciénegas is the name of this place. It actually translates into the four marshes. And it's been proposed as an ancient ocean, the remnant of an ancient ocean, part of the Protogulf 100 million years ago. So when Pangaea was splitting up and the Gulf of Mexico was being made, actually this particular area right down the middle of Mexico was actually part of the Proto-Gulf. And this was done in a paper with my collaborator Valeria Salsa at UNAM, and it was in PNAS. And the thing that's remarkable, and although I have my reservations about it, I'm going to show you some information that's going to make it appear that not only can you say this geologically, but it looks like biologically there's remnants of this marine ecosystem that have come up in the waters that are coming down from underneath. The other thing that's extraordinary about this place is it's low in phosphorus. Now, I want you to think about that for a minute. The total phosphorus ranges between 0.05 and 0.4 micromolar, with most of the poses in the 0.1 to 0.3 micromolar range. Now, what do you know about phosphorus? If you ever think about limiting elements, we've talked about, probably you've heard about nitrogen being limiting. I mean, you have to have fixed nitrogen. The planet's biology has to have fixed nitrogen. So if you don't have fixed nitrogen, it can be limiting. That's why you have to put fertilizer on your gardens. But what about phosphorus? What happens if you're low in phosphorus? And this is very, very low, especially for any other terrains that are similar to this. What do you know about phosphorus? What's phosphorus used in biology? DNA, RNA. It's also the ATP. It's the currency. Pretty important element as far as biology is concerned. So, Arizona State. Oh, let me tell you this. This is one other thing that's especially interesting about this place. It's Galapagos-like, and basically what they have discovered is that the diversity of endemic species, so species that are not found anywhere else on the planet, just here. They've already described about 70 of them, and that's equal in diversity to the Galapagos Islands. So basically what you have here is where the Galapagos is actually isolated because of being surrounded by water, and life has been able to evolve and adapt there independently of anything else. You have a similar situation here where the isolation is because of that big mountain range that circles the valley. And it's proven in this fact that we have over 70 endemic species there, ranging from reptiles, fishes, scorpions, insects, plants, all different kinds of things. So why do you have, in the pools at Cuatro Ciénegas, stromatolites dominating? This is an example again of the Australian Shark's Bay ones. This is a little bit what they look like when you look in the fossil record in the Pilbara. She got on to me the other day because I said Pilbara. It's Pilbara region. And so this is what some of the different morphological types look like at Cuatro Ciénegas. So this is that aerial photo that you saw. I call this a shelf stromatolite. But I mean, you can tell how, to me it's elegant. This is made entirely by microbes, basically, lithifying, pulling the carbonates out of the water and encasing themselves in it. This is another type. This is just sort of like this is a pool that has a spring at the bottom, about 40 foot. So it's very quiet and tranquil, not much going on. These you actually find in a river that flows across in one area of the valley. And these are like, being from Texas, I call these kind of like microbialite tumbleweeds, because they actually find something to nucleate around, and the microbial community nucleates around that and then just grows yearly, and it can roll around on the bottom of the river floor. This is us actually trying to take a sample from one of the more domal stromatolites. And then I just put this in here. You're going to see this again. I just think this is too cool because I'm going to talk about what this community is made up of, which one of the things that it's primarily made up of is cyanobacteria. And if you know anything about cyanobacteria, you know that they're the only things on the planet that make oxygen. The only thing on the planet that makes oxygen. The reason plants make it is because they actually co-opted the ability from cyanobacteria. And this is a mat. You can clearly see the green cyanobacteria. This is in Cuatro Ciénegas when we were swimming underwater. And these are oxygen bubbles, where the oxygen is actually being produced. That's just cool.
So in that upper right, is it sort of crusty if you feel it? I mean, what's the nature of it?
Okay, so that's actually another great question. If you took your foot and you punched it like this, it would just fall apart. It would disintegrate underneath you. If you stuck this into it, you could probably stick it in and it would kind of crumble around it. But if you touch it, it feels hard. And the thing that's a little bit confusing to me, everybody that I've talked about that looks at sort of current stromatolites, even the one in the Bahamas and Shark's Bay, they all kind of have this structure. I don't know if you can see this. This doesn't look like, when you look in the geologic record at stromatolites that people call stromatolites, they're very, very distinctive layers. This kind of reminds me of like tiny broccoli florets. And that's kind of what it looks like whenever you section it. This is a section. This actually looks like that. And if you took a piece of this, it would look similar to this too. So it's crusty. a little harder than crusty, but definitely pretty fragile. And this is the ones that are alive. If they're alive, they're like that. There's actually a pool down there that has some dead ones in it, and they're hard as rock. And I know they're dead because there's nothing, there's you don't see any kind of, if you open them and do any kind of microscope work, you don't see anything in there. I mean, they're clearly, they've turned into rock.
Would you say you're puzzled as to how the strata from the old ones are
Yeah, so the story is you get the strata because you have some sort of seasonal input, right? But so far, there's a little bit of strata. If you looked real close, you would see a little bit of layering up here. You would see a little bit, but nothing like what you see in the geologic record. I mean, this you have to. I mean, you'd really need a rock lens to look at this close. I don't think the geologic record, the stromatolites in the geologic record, you don't need any kind of magnifying aid to see them. So I'm a little confused about that. And I actually talked to Abigail Allwood, and she's seen the stromatolites in Shark's Bay. She's the one that did the paper on the Pilbara region, the geologic, the most recent paper on the stromatolites in the Pilbara region. And she says the Shark's Bay ones are like this too. And that's a completely different ocean dynamics there than it is here, but it's basically the same. So I'm confused by that. I don't know what to think about that. Anybody have any thoughts, or we?
I just have another question.
Sure.
Just on the same vein,
Yeah.
How thick are they, and are the microbes just at the?
So it's not like just a layer. I actually have one at home in my aquarium. It's not just like a solid layer. It's like, think about taking a piece of broccoli and sticking it out there and putting carbonate all around it. That's kind of what it looks like to me. I mean, if you look down deep into it, you can see that there's little green cyanobacteria down in there too.
Oh, so there's stuff living in the middle.
Yeah.
Most of it is living.
Yeah. And there's little snails all around that. There's 4 or 5 snails that are eating on every one of those, like they call those round ones oncolites. They call this one oncolites. And if you look real close, you'll see a little snail here, and there's always a worm in there somewhere too. Now, I don't know how well you can see this, but I don't really want you to take anything home from this. This is a figure out of Abigail's paper. And this is where she basically went to the Pilbara region and she categorized all the different morphologies of the stromatolites there. And I remember one time when I was down at Cuatro Ciénegas looking at all the morphotypes I just showed you and thinking, what would this look like if you preserved this in the rock record? Well, see, she's got basically sort of very similar morphotypes that she's cataloged here as we see down there. The oncolites, the big shelf ones, the domal ones. I mean, even in that small valley, we've got several different morphotypes of stromatolites down there as well. So this is actually, go ahead and advance all these. These are the 3 particular places in the valley I'm going to talk about. This is the valley shaped kind of like, I love my Mexican collaborators. They're very eloquent in their language, and they say that the valley is shaped like a butterfly, which it sort of is. And I'm going to talk about 3 places. I'm going to talk about the Churince system, which is basically starts out as a spring, meanders across the desert valley, empties into a lagoon, and there's an evaporative process that goes on. Then meanders across the valley again and ends up in a big evaporative lagoon, and that's where those gypsum dunes are forming. We're going to talk about the Rio Mesquites, which is where it's just an above-ground kind of fast-moving river, and that's where those round tumbleweed oncolites were. And then I'm going to talk about the Pozas Azules, which is where those big shelf stromatolites were. Okay, so what's I hope you think it's a pretty neat place already, but why is it so special? Well, the theory is that because of that low phosphorus content, remember I told you there were snails on those stromatolites, those microbialites. Supposedly, because there's low phosphorus there, this was a paper done by Jack Farmer and Ferran Garcia Pinchel out of Arizona State. They basically measured snail poop and growth of the stromatolites and measured the amount of cyanobacteria, and they determined that the cyanobacteria were growing just a little faster than the snails could eat. And that's why the paper actually says that that's why there's stromatolites dominating this area. And that's probably, their supposition is because of the phosphorus content is so low. Even though you have complex organisms there, predators, they're just not robust enough to keep up with it. And so that's always been considered in an astrobiological context as a proxy of early Earth, right at the transition from prokaryotic to when you begin to have eukaryotes being dominant over prokaryotes. The low P is the limiting nutrient. It's interesting because I've told you about the marine origin issue, and it has active lithifying mats. So that's the reason why we study it, Cuatro Ciénegas. Okay. So now I'm going to get into a little bit of the actual work that we're doing down there. And we actually went down there for a specific reason. And the reason was we wanted to understand how genomes, prokaryotic genomes, actually coalesced. Because at the heart of understanding how the origin of life or how life is adapting, you have to understand how the genome comes about or how it continues to adapt. And the thing that's interesting about that place being dominated by prokaryotes, basically prokaryotes don't get to have sex. Their way of actually creating diversification is through horizontal gene transfer. That's their major tool for prokaryotes for genome dynamics. All right. So I'm not going to go over this too much, but I know you've all seen. How many of you have never seen this tree? Okay. Clearly, what this is what this is a visual aid for is there was a time when I first started working. Vicki mentioned that I got my degree with George Fox, who was one of the co-discoverers of archaea. They did it because they didn't believe there was any gene transfer going on, that you could actually take a molecule from today's life and you could trace it back and figure out what the relationship was here. So about 15 years ago, when there was so much sequencing going on, people began to realize that there was a lot of horizontal gene transfer going on, and it scared everybody. Well, if there's this much going on, how could we ever understand anything about how things are related in the prokaryotic world? And this is Ford Doolittle's sort of example of just how complex all that horizontal gene transfer may have made us understanding how life is related to each other. This is an old slide, and you don't need to know anything about it except you need to recognize that this is 15 or 20 bacterium. This is the length of their genomes, and the blue is their own DNA, whereas the yellow and the pink represent different amounts for each one of those bacteria of alien genes that were located in their genome. So what you need to get from this is just one thing. It really doesn't matter how long your genome is or how big your genome is. Different bacteria will accept different amounts of horizontally transferred material. Okay, so now I need you to think for a second. So I've told you this place is low in phosphorus. So when I heard that it was low in phosphorus and I was thinking about horizontal gene transfer, a couple of my friends and I, we got together and we said, well, if you have a place that's low in phosphorus, is that going to affect horizontal gene transfer? Well, you need to know a little bit about how horizontal gene transfer actually happens. And it can happen three ways. And you should be familiar with these if you've done anything in molecular biology because we use these all the time. Transduction, which is viruses. So a virus infects the host bacterium, and when it does, it brings a little bit of DNA from its previous victim in. That's transduction. It could be conjugation. This is the only thing that bacteria get to do that sort of resembles sex. They get to get next to each other, and they need to exchange plasmids or different kinds of other DNA that's within them. Or transformation, which means that a cell just breaks open, and the DNA is just lying around in the environment, and for some reason, this host bacterium has the ability to actually suck it up into it. Okay. I'm going to see if you can figure out what we figured out. If you know the place is low in phosphorus, and you're trying to understand how horizontal gene transfer happened, do you think an environment low in phosphorus would favor one of these vectors over another? Just think about it. What would you think? This one, would this take a lot of phosphorus or not much phosphorus? Got to make a lot of viral particles. They're all RNA or DNA. So what's this going to do? This one would probably take a lot, right? This would probably take a lot of phosphorus. This one, we're just going to let them enjoy themselves. We're not going to talk about it. They could have sex there. Transformation, we're talking about this is just DNA out of the environment. If you don't have much phosphorus, wouldn't you find ways to scavenge this up and pick it up? I mean, if the environment's low in phosphorus, if you could adapt to pick this up out of the environment whenever something's broken open, that seems like that would be the best strategy. How many of you think that sounds reasonable? Okay, I did too, but it's not right. Turns out, this is what we thought. Mechanisms of gene exchange, transformation. If you have low P, transformation's probably going to be the best thing that you're going to do. Transduction, the viruses will probably be the worst thing that you could do.
I thought conjugation should be good in the sense that it's most efficient. You're not just spraying your phosphorus around.
That's actually good. There's a problem with conjugation. One thing I didn't go over with those three things, each one of them can carry different amounts of alien DNA, and they have to carry things that are more related. So if you're actually conjugating, you're actually probably more related. So that's more inter-specific type. Not likely to bring in something that, is just completely foreign, which, I'm a biologist. I like the really odd things. Okay, so it was about this time when somebody told me about Cuatro Ciénegas, and I was thinking about phosphorus, and I was thinking, okay, should I shift focus from microbial, I could be a microbial ecologist, and I have to admit we sip a little tequila in the desert when we go down there, or sit at a computer in Houston and analyze those ACGs and Ts. So I tried to become a microbial ecologist, which turns out to be very complicated. So we began this suite of projects at Cuatro Ciénegas to try to get at what was actually happening in those microbialites and in the water and in the poses. So this is just sort of, I showed you all those people before. These were some of the things that we actually began to do. So this one I'm going to talk about, bacterial and viral metagenomics of the microbialites. So all we did was we went down and took samples. And I was telling some of the students at lunch about one of the horrendous things that happened when we took a sample. Took a sample, we take it back to the lab, and we don't care. We don't try to culture anything. We don't try to separate anything. We just take the whole sample back, extract all the available DNA in it, and then sequence it in little bits on a 454 sequencing machine. So rather than think about, like an individual microbe, we actually took that whole stromatolite, took it back to the lab and sequenced every piece of DNA in it to try to figure out something about the community rather than the individual in the community. Okay, the other thing that we did was we did an isotope analysis of cores and sediments and the microbialites. There was a bacterial census done over the whole valley. Genome sequencing of a specific bacterium. We did ecosystem nutrient perturbations and analysis. So in other words, stream-side buckets filled with those little oncolites and dumped phosphorus in it to see what would happen, and then ran analysis. Experiments to determine how fast those microbialites were forming, continuing to isolate new species, and then comparative census of microbial communities. So I'm going to talk about three of these right now. I do want to tell you one thing before I continue. We are very privileged to actually be working at this place. It's now under the Cuatro Ciénegas Valley is actually a World Heritage Site now, and it's protected by the Mexican government. And as I told you a minute ago how fragile those things were, you can imagine those beautiful shelf ones if there were very many people swimming around, which this is a place to go at Easter for a lot of northern Mexican people. You can imagine how fast that would be decimated. So one of the ways that we pay we try to show our respect and our appreciation is we actually take a planetarium, a portable planetarium down. These communities don't have any movie theaters. Most of them don't even have television. And so we have 3 planetarium shows that were translated in Spanish. So we take these down about 3 times a year to help educate the community. So the one thing I want to talk about is the bacterial census that we did of the valley. Okay, so I told you about the endemism of the macrofauna. I told you there were over 70 endemic species. Well, we wondered, is it possible that you might have endemic microbes here as well? Now, that's a problem. If you're a biologist, you already know what the problem is. We don't really know what a species is. We don't know what to call a species in microbes. There again, it's that problem of not having sex. They're clonal, so we're not real sure how you say a species. But let's say that we could find an example of such an extreme case that we had to say the microbes were endemic to that region. We began to think about looking for that. We also wanted to see, well, just what are the microbes like? This is in general in the water systems down there. And this was a census that was done. And all you need to see from this is that no matter where we took them, you see this big blue section? Those sequences for the microbes that we took down there were all more closely related to marine sequences in the databases that are held all over the world than they are to anything else. Now, when my collaborator Valeria told me that, I thought, it cannot be possible that 100 million years we still have some sort of marine signature in the bacteria because it was left over from the protogal. Well, but they did several sort of statistical analysis to make sure that this wasn't some sort of misleading result, and it turns out that it looks true. It looks like the majority of the residents there are more closely to other marine organisms than they are to anything else. So this is what I was talking about. We thought, well, so they're more related to marine organisms, but are they endemic to that region? So I want to talk a little bit about that Chirense system. Remember, I told you that it started at a spring, meanders across the desert floor and goes into a lagoon, and there's some evaporative process, and then this is the big evaporative. And here's the big evaporative pond there. And you can see there's a variation in magnesium and sodium and calcium going from one end to the other. So it turns out that when you look at this, when you look at that particular region, you find a high percentage of bacillus strains there. Not only that, we found one in particular. Let me talk a little bit about bacillus. It turns out that we were focusing when we first went down there because the stromatolites were so intriguing. Everybody talks about stromatolites being produced by cyanobacteria, and they do have a role in that because they are a primary producer. But it turns out that cyanobacteria don't like to be isolated. They don't like to be in pure culture, and they are terrifically difficult to get into pure culture. So we began to look for other organisms there that were indicative of the region that might be easier to cultivate. It turns out bacillus are. So they were widespread throughout the valley. They're in different environments. We found at least 17 species in that little Chirense system that I showed you. They're fast and easy to culture. And for another project, they're actually important in nitrogen cycling. I'm not going to talk about that. But so lo and behold, we found Bacillus qualwiensis, and we sequenced the genome. Turns out that it's the smallest Bacillus genome known, only 3.3 gigabases. Now, bacillus are usually sort of, in the world of bacteria, they're kind of complicated because they actually produce spores. So they usually have big genomes. So it was very unusual to recognize that this one had a very small one. And this particular group, you find relatives of this, not this one, but relatives of this all over the world in marine and saline environments. So in the sequencing of it, this is just your standard. This doesn't mean anything. I don't care that you even look at this. This just proves to you we actually sequenced the genome. This is but this is important here. So it turns out that this little bacillus that's down there in Cuatro Ciénegas has the smallest genome, has actually co-opted a myriad group of biological mechanisms into its genome. While it's been reducing it, it's actually co-opted through horizontal gene transfer some very interesting attributes. One of them, from the cyanobacteria around there, it's taken sulfolipids. Now, let me explain that to you. How many of you know what a membrane, a cell membrane, is made up of? Has something in it we've been talking about. Phosphorus. It's phospholipids, right? What have I told you about this place? It's low in phosphorus. So guess what this thing's figured out how to do? Use sulfur. It doesn't even make phospholipids anymore. It makes sulfur lipids in its membranes. And it co-opted that from cyanobacteria with the rest. So a horizontal gene transfer event that we actually know occurred because of it being in this place. Now, I won't go over too many of these because they're not as important as that is right now, but what I'm trying to tell you, this is likely, this particular bacillus that can do sulfolipid metabolism is probably not present anywhere else in the world because this unique environment. So this is probably an endemic bacterium from this region. I don't know what that was. Tell me it's time to stop. Okay, so it's the first described heterotroph to produce sulfolipids, even though relatives of it are common throughout the world in saline and marine environments. It's a perfect candidate for analysis of those globally distributed relatives to understand what endemism might mean in the bacterial world. And this has actually been accepted by PNAS. So, okay, the next two slides, I don't want you to get, the next one's going to be very complicated. And it's just to show you something. You know, I tried to get you to think through the process about what we thought about horizontal gene transfer happening in a low phosphorus environment. And about a third of you agreed with me that this probably seemed reasonable. So this is actually what we ended up with. Just like all in biology, it's always way more complicated than you ever thought it would be. It turns out that we knew that we had low P, and we know we have high diversification. Remember, I told you this is like Galapagos. Even in the microbial world, there's a lot of diversification. And it turns out that low P affects everything. It doesn't just affect one. It affects everything. There's low transformation, low conjugation, low transduction. And actually, interestingly enough, we believe that leads to kind of a high diversification that happens in the independent little lineages all throughout the valley. Now, the selection for P actually does have some consequences on a genome. It actually probably forces any genomes down there to purge themselves of any genes they don't need. Remember I told you this had the smallest genome of any bacillus? And so there's probably two things that happen in a low phosphorus environment. You have extreme diversification because of a local adaptation, and you actually try to get smaller and smaller genomes. Okay, so putting it all together as far as endemism and horizontal gene transfer at Cuatro Ciénegas, the dogma has always been that with respect to bacteria, everything is everywhere. No matter where you went on the planet, every species of bacteria is there. It's just not prominent. But at Cuatro Ciénegas, everything is not everywhere, and maybe in some places everything is special. And so we actually think Cuatro Ciénegas, because of the local adaptation in its Galapagos-like environment, has actually, not only does it have macrofaunal endemic species, but it's also bacterial. And this is actually a Nature Reviews in Microbiology paper.
So,
yeah, sure.
Catch how you know the microbes are affected?
Well, no other bacillus in the world, of all the 50 or 60 that have been sequenced, none of them produce sulfolipids. Now, you actually make a good point. I mean, it's a bacillus, right? I mean, and it's a bacillus, it's related, it has other organisms it's related to. So you have to define in your own mind whether or not you want to count a microbe as being the same if it has the same ribosomal RNA, or whether or not it can do unusual things. So it's a matter of definition, but I think if you want to count something as endemic, this is about the strongest argument you could have if you're talking about microbes. I'm okay if you want to disagree with it, because I can understand it. But nowhere else, it's never been reported that any other bacillus, not only any other bacillus, but no other heterotroph makes sulfolipids. Sulfolipids are made in the ocean all the time by all kinds of cyanobacteria. That's well known. Okay, so now I'm going to talk just for a few seconds about the metagenomes. We actually took one of the oncolites and a piece of the stromatolite, and we did the metagenome study. Remember, that's just the whole community RNA. And we looked at the viral component of it and the bacterial component, and we also evaluated the isotopes of the water in the microbialites. Now, this was quite a project. Those two sentences make it seem real simple. I'm going to go over the viral community. I only have this one slide. This was a paper that came out in Nature about 2 weeks ago. And basically, this was kind of scary to me, the results of this, because what it actually shows is Rio mosquitos is the oncolite. This is the shelf stromatolite. And these are each different marine samples of virus particles, right? So these are viruses from this, and then these are all marine. And all you have to know is that if you have anything that's greater than 0.25 over here, that means that whatever is greater than 0.025 has a greater difference, wait a minute, let me get this right. In samples, yeah, okay. This is comparing to the combined marine sample. And if there's no difference, if it's less than 0.05, then it's exactly like it has a relationship to the marine sample. So it turns out these are all marine samples, and if you compare the Gulf of Mexico viral viruses with the combined marine viruses, there's no difference, right? You following me? Posus azulis, that shelf stromatolite, looks exactly like a marine sample. So in the paper, this actually corroborates that idea that not only does the bacteria look like it has a marine signature, but the viral components look like they have a marine signature as well. Building on the argument that this is a relic of the ancient ocean. Now, I love this picture just because this shows how dirty this work was. When we were doing the isotope analysis, when we went down to get the samples to do the isotopes, this is David Hollander, and he actually looks like that most of the time. And this is us taking a core. This is Maya Breitbart and David, and this is the core. And so David is the one that does all the isotope analysis. This is the bacterial component of the stromatolites. And this is just to show you the light bar is the shelf stromatolite, and the dark bar is the Rio mosquitos. So looking at the bacterial fractions, we tried to figure out what were the components, what were the microbial identities in each one of them. And you can see one thing very clearly. They look like there's two different communities there, right? They're not the same. Does that seem reasonable to you? They're different. Okay. So the only thing you need to recognize about this is that if you compare the this is a slide of the functions of the genes that we found in the metagenomes. So these are all the different categories. And the comparison is the microbialites compared to marine ones, to marine functions, and compared to freshwater. And the only thing you need to notice, the only thing I want you to notice on this is that this little red bar here is sulfur metabolism. And it actually shows that no matter whether you compare it with marine bacteria or freshwater bacteria, the bacteria in these microbialites actually have something's going on with the sulfur cycle that's not normal for most of freshwater or marine biology, microbiology. So there's something about the sulfur that's causing Both communities, no matter what organisms that are in them, both communities have a high sulfur cycle. There's something going on about the sulfur cycle. This is just another graph to show you a little bit about, remember I told you you're going to have lithifying mats either because you've changed the metabolism or you've done something about making the extra polymeric substance. And this is showing you that both in the round stromatolite, the microbialite, and the shelf stromatolite, there's actually a lot of EPS production and a lot of degradation. So extra polymeric substances are also active in this. So these microbialites are probably using sulfur metabolism as well as creating EPS to actually cause that precipitation to occur. So basically what these slides are showing you is that model that Dupraz had actually put forth is actually corroborated by this evidence. There's at least two. The EPS and the metabolisms are actually affecting the fact that carbonate is actually precipitating on these communities differently than what it would in other places. Okay, so basically Stroms are complex, redox-dependent, highly dependent communities. They're highly integrated. Their metabolic composition is distinct from other marine and freshwater microbial communities, even though the taxonomy of the two Stroms are very different. Their metabolic potential is the same. And that particular potential is important for carbonate precipitation. The isotopes, I'm not going to go over that, but the isotopes indicate that heterotrophic respiration occurs of the photosynthetic biomass. So even though the cyanobacteria are there, and they're actually the primary producers, their metabolism is not the one that's actually causing the precipitation. It's the sulfur and the heterotrophs that are actually causing the precipitation. The sulfate-reducing bacteria are key components to the community. Now, what does that have to do with astrobiology? That has to do with astrobiology, because we are beginning to understand what the constraints are that allow precipitation to happen and for you to get a lithified mat. And if we can understand the constraints of what has to happen in order for you to get lithification, we can understand how, through the rock record, what the ocean chemistry had to be like or what the microbial community had to be like in order to get lithification. And if we can understand that, we might be able to actually extrapolate that for if you were going to look for life stromatolites, if you could actually visually ever look at something else to see what evidence of stromatolites might be on other planets or microbialites, you would know what the conditions had to be. And that's actually getting us, that's actually a step forward. We're going to actually be able to measure horizontal gene transfer using the Cuatro Ciénegas isolates. We'll understand a little bit more about genome coalescence. I mentioned the lithification of the ecosystem. And then this is important. We'll understand and be able to evaluate P as a limiting nutrients and its effect, which can provide better early Earth model constraints. And this is something I talked about at lunch. This may seem kind of, it's just advanced life on Earth that we're actually talking about. But the fact of the matter is, no matter where you are in the universe, you still have the same periodic table. So the same element building blocks have to be used no matter where you are. So the fact that you're precipitating just a chemical process, carbonates out of water by biological intervention, could happen anywhere in the universe. We wouldn't have to actually understand what the life was like. So this is just two more shots of Cuatro Ciénegas to show you just a little bit more about how beautiful it actually is. I mean, this is just how majestic the mountains are that surround it. And I'll give my thanks to Vicki and the rest of you for listening.
Questions for Janet?
Through what you said about low phosphorus affecting rates of transduction, could you extrapolate that back and say anything about rate before the gene transfer and other environments?
I mean, you could say that. I don't know that we've got, I think that's too. I think that's putting too much in what we have at this specific place. I don't know how much. What I showed you, actually, I mean, you have to appreciate that microbial ecology, very seldom do you have a place that's isolated like this, that's dominated by microbes. So this is a really unique situation. And I don't know, even though I think it has some relevance for general principles, I'm not sure what, I mean, it's too early for me to even begin to think that. But I mean, we can have fun talking about it.
I had rather expected that the precipitation of carbonate would be if bicarbonate was being used as the carbon source. You pull out one CO2, you're left with carbonate, and I can imagine that if it's a single individual cell, carbonate might precipitate away from it, but if you had a mat of blue greens, that maybe the calcium carbonate would kind of sit on top of it. You didn't refer specifically to that reaction, so maybe there are other things going on?
That reaction does happen when they create the EPS. It actually does sometimes cause the bicarbonate to split and that actual reaction to occur. I don't know specifically if that's happening here. I don't know that, but that does. I just didn't go into that much detail about it. But I mean, that certainly can happen. I mean, the basic thing that you can take home is something has to happen locally for that precipitation to occur. And as much as we can understand about what's causing that local precipitation to occur, whether it's pH change or bicarbonate ions, breaking the bicarbonate up or whatever, I mean, that adds to us understanding how, because you can go to places that it doesn't happen.
Do you have a sense from your metagenomic sequencing how many organisms possess?
No. Yes. Okay, the question was how many other organisms at Cuatro Ciénegas might be utilizing sulfolipids in their membranes as the bacillus is doing. That is an excellent question. And if I could ask for a reviewer for a grant, I'd put your name on it. Because that's a grant we currently have out for exactly that thing. I mean, you would think that probably there'd be lots of stuff down there that's resorted to that strategy. I mean, why other things do. That's an excellent question.
Do we know the reasons why phosphorus is so low there?
No.
Would that be evident that that's a remnant of very, very ancient, sediments? Thinking that we don't still know how phosphorus was somehow oxidized first to developing everything.
Right.
Maybe this is an answer to that.
Okay, I'm going to have to admit, I am not a geologist, and I know there's no way I could answer that. Maybe somebody in the room here understands a little bit more about phosphorus availability and what happens, when you're looking at an environment and there's a lot of evaporative processes going on. I don't know what would make phosphorus low there. I have no clue. Let me ask you a question. How many of you in here believe that's sort of a relic of ancient oceans? You think that's reasonable to think that? You're okay with that? Usually I get some very, pointed arguments about that. I don't understand how you could do that. I don't understand how it could be, the biology could be, ancient relics for that. The problem is, we keep having data that shows that. I mean, I didn't tell you that there's diatoms down there that look like they're marine. I mean, with bacteria, I could have said, well, maybe it's sort of convergent evolution because it's in a salty environment, although it's not sodium chloride. But it's a marine signature. It's not a salt signature that's showing up. So y'all are much more. Maybe that's people that live in Seattle are much more willing to think outside the box.
Don't the process have very different compositions as well? I remember when we first went down.
Yeah, so it's kind of some of them you can have one that's like real big and it's about this much space between the next one, and this one will be dark brown and that one will be blue green, and there's about this much space between the two. But I don't know what that translates into. There's some chemistry differences. But I don't. We've not been able to correlate anything with that.
So I've got a detailed analysis of the
Mm-mm. There's a lot of projects.
We need money, and I'm sure Janet wouldn't mind
Oh.
a rotation.
Oh yes, we always have. We love to have.
Anybody would like to go on a field trip with Janet at some point? So
I'm thinking about asking Mike Rowe to come down. Do you know Mike Rowe? Y'all don't watch Dirty Jobs? Oh my gosh, we have to dig around in the sulfur smelling,
Yeah. It's pretty dirty and dusty. and you need a crash helmet for the ride across the desert because
we've destroyed
get
2
thrown
cars.
against the car. Anyway, do we have any questions on VideoCon?
No.
No. Okay then. All right. Are there any more questions for Janet? Yes, John.
So what kind of cyanobacteria are there?
So there's Calithrix.
And the other question is, how closely related are the genes for the sulfur lipids in those cyanobacteria to the bacillus that you got? How closely related
Or the ones locally?
Yeah.
Okay, that's a problem. Calithrix is there's a succession of I guess it's just like anything in biology. Something moves in, and then other things move in. So clearly, Calithrix, which is a cyanobacteria that has this real heavy brown, you can almost see it with the eye, sheath around it. It's clearly sort of the thing that's on top of all the surfaces of the microbialites. And it's been impossible for us or the Arizona State people to isolate that. So we have no clue how similar those sulfolipids are to the ones that might be resident there. Now, actually, I should look in, because we should have some information on that with the metagenome. We should be able to tell that. And I don't remember that we ever looked at that. That's actually a good point. So I will look at that and see. But we would not be able to, even at that point, we would not be able to identify it with a particular species, I don't think, because we don't know we don't know which gene might be with which species there. That makes sense?
Yeah.
Okay.
I thought with the metagenome.
We should have been able to figure it. We should have been able to see that. I'll have to go back and ask that in the data. I don't remember.
The same question is what the overall diversity of the genes
right.
And sulfur lipids are. Do they extend beyond the bounds of the bacillus type?
Well, I mean, there are no sulfolipids in bacillus, so there's no way to compare them to other bacillus.
No, I mean, within this environment.
Yeah.
How diverse is that gene?
Well, definitely. I'll have to go back and look at that. I don't know. I actually don't know the answer.
And bacilli, of course, are one of the original weeds for doing transformation experiments at gram-positives. They are also fairly closely related to cyanobacteria on phylogenetic trees and should potentially be compatible with transformation.
Well, the first time that the sulfolipids were described was with Obed Deja's, I can't remember the name. Anyway, they described them in the Sinococcus and Prochlorococcus that are all in the temperate oceans. And they've actually done metagenomes of the oceans, and they have found what they think is a heterotrophic sulfolipid component, but they can't figure out where it's at. I actually read that the other day. So, I don't know what that means. They're not saying they're bacillus. I mean, I don't know if bacillus are in open ocean too much, but, of course, the open oceans are low in phosphorus, right? That's why the Sinococcus and the Prochlorococcus have co-opted that ability. They sometimes, when the phosphorus is plentiful, they make phospholipids, but when it's not, then they revert to sulfolipids. This thing doesn't do anything but sulfolipids. It can't make phospholipids anymore.
All right then. I think we'll thank Janet again for her presentation.